splitting. And so instead of having 2 duplicated chromosomes, you end up with 4 unduplicated chromosomes. Now, nuclear division is just the first step of the cell dividing. What happens next, where the cytoplasm divides? What's that called? Cytokinesis. And so when cytokinesis happens, each of the daughter cells will get 1 set of the unduplicated chromosomes. So that's why the chromosomes have to duplicate. This chromatid right here is eventually going to break off when the centromere splits and become its own chromosome going into one daughter cell. And this centromere with its chromatid is going to break off and go down to the other daughter cell. And so the daughter cells end up with the exact same number of chromosomes that the mother cell had to begin with. You start off with 2 unduplicated chromosomes, and then each daughter cell ends up with 2 unduplicated chromosomes. Alrighty? All right. Well, let's see now. There are certain times when a cell wants to divide it as rapidly as possible. And a good example of that is the zygote. As soon as the zygote is formed at the moment of conception, it only has 9 months to form a newborn baby with around 4 trillion cells. And so that's going to take a lot of division going on constantly and very quickly. And so you would think at first that if a cell was interested in replicating as quickly as possible, duplicating itself as quickly as possible, it would just go divide, 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 divide. But what they found is that even if a cell is interested in reproducing as quickly as possible, it can't just go divide, 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 divide. It has to have a resting period between divisions. And that resting period is called interphase. And I guess resting period is not really a good description because the cell is not resting during interphase. It's preparing itself. It's making preparations for the next round of cell division. And so when cells are reproducing as rapidly as possible, what they do is the interphase, where they're resting and preparing for the next division, then they do a round of division, nuclear division, followed by cytokinesis, and then the daughter cells then repeat what just happened before. They go through interphase, and then another round of cell division, nuclear division, cytokinesis, and then those daughter cells then rest, they go into interphase, and then they do their next round of cell division. In a lot of textbooks when they're talking, and this process is called the cell cycle, since the cells are cycling through interphase, then cell division, then interphase, then cell division, then interphase, then cell division. And it's usually diagrammed in textbooks like this as a clock, because just like the clock, the hands of the clock sweep through the same hours again, day in and day out, over and over again. The cell is sweeping through interphase, and then cell division, and then interphase and cell division, and interphase and cell division, as it goes round and round through the cell cycle. And incidentally, in this diagram that I'm going to be showing up here, the purple parts of the diagram are always going to be the interphase when the cell is not in the process of dividing, and the white parts are always going to be the division parts, mitosis, nuclear division, and cytokinesis. All right. What they've found by studying the cell cycle is that the cell division part, let me fill that part in, there we go, is the mitosis, nuclear division, followed by cytokinesis, you know that. But what I was going to say is that the interphase part is subdivided into different phases. They're called G1, S, and G2. And the G stands for growth, because in G1 and G2, the cell is preparing for division by growing. The cytoplasm is getting larger, the organelles are being duplicated. In other words, if the cell starts off with 7 mitochondria, it'll eventually grow to have 14 mitochondria, or if it has 2 Golgi bodies, it'll end up with 4 Golgi bodies. It's just all about more cytoplasm and more organelles in the growth phases. But the S part of interphase is, the S stands for DNA synthesis, and that's where the DNA replicates. That's where the chromosomes duplicate is during the S phase. All right, so I'm just going to not show the word interphase there, but the purple parts in the G1, the S, and the G2 are the interphase parts of the cell cycle. The part where the cell is actually dividing, mitosis, nuclear division, and cytokinesis, the white parts right here, are sometimes referred to as the M phase of the cell cycle. I guess because it starts off with mitosis, but the whole thing, mitosis and cytokinesis, are the M phase. The mitosis part, the nuclear division part, is itself subdivided into 4 phases, and they are in order called prophase, metaphase, anaphase, and telophase. The 4 parts of mitosis nuclear division. Now, in your textbook and some other textbooks, they actually stick in a 5th phase of mitosis between prophase and metaphase, and they call that prometaphase. And I guess I don't agree with that. It seems to me that the description of prophase and metaphase are perfectly fine without trying to stick an extra phase in there, so I don't do that. But be aware in your textbook they will mention a prometaphase. All right, well, what I want to do is go through the complete cell cycle, all these phases, and talk about what exactly is going on in the cell at each of those. And let's see now. Let's start with G1. So there's the cell. There's the membrane right there, and these are the organelles, and there's the nucleus. Why do the chromosomes look funny? Who can tell me that? Why do they look fuzzy, I should say? There. What's the name of that, where the chromosomes have come off the histones? They're spread out. Chromatin, right? The chromosomes are still there. Matter of fact, you can kind of see the centromeres there. But during interphase, the DNA of the chromosomes partially unwraps from the histones, and so the chromosomes are not condensed tightly, that their DNA spreads out all throughout the nucleus, and we call that the chromatin state, or you could call them uncondensed chromosomes. So under a microscope, you would not be able to see individual chromosomes. The nucleus just looks like this blob of DNA, but all the chromosomes are still there, they're just spread out. And that's actually the state of the chromosomes throughout all of interphase, G1, S, and G2. They are chromatin. It's only here during mitosis that the chromosomes actually condense. Okay, so G1 is the first growth phase. It occurs right after the mother cell did cytokinesis. In other words, right after the 2 daughter cells are created. And as I said before, it's just growth. New organelles are being manufactured. More cytoplasm volume is being added. The membrane of the cell is growing also to accommodate the cell getting larger and larger and larger. Now, since the chromosomes are in the chromatin state, you can't really see them, but let's cheat for a second. If you could see them, they would be unduplicated because the chromosomes duplicate here in S phase, and we haven't reached that yet. Okay, let's put them back. There we are. All right, so now we're going to enter, the cell enters the S phase of interphase, and this is the phase where the DNA replicates and the chromosomes duplicate. So this is the phase where DNA polymerase III and primase and replication forks and origin of replication bubbles, all this stuff that we talked about happens. So you go from each chromosome being unduplicated to duplicated. And again, you can't see it because the chromosomes are in the chromatin state, but if you could see it, this is what it would look like. All those enzymes and replication bubbles and replication forks would do their thing, and chromosomes would get duplicated. So, but you can't see it. And then lastly, G2 phase, the final growth phase before we actually start getting some division. More growth, so cytoplasm gets even larger, more organelles are generated, the cell's membrane gets larger and larger to accommodate the larger cytoplasm. All right, now we finally reached the beginning of mitosis, so we're about to enter prophase. What I'm going to do is make the organelles vanish from the diagram, not because they vanish in real life, but just because it makes the diagram a little bit more clear if we get rid of them. In your lecture handout is a table that shows the 4 phases of mitosis and what's going on in each phase with the chromosome, the nuclear membrane, and something called the spindle apparatus. So let's see those guys. All right. When the cell goes into prophase, that is when the chromosomes condense. The DNA starts winding more and more tightly around the histones, and so you, under a microscope, you would start to be able to see the chromosomes appearing. The nuclear membrane starts to break down. Why? Well, remember that the chromosomes are going to break apart and start moving towards opposite poles of the cell, and the nuclear membrane would just get in the way of that process. But maybe the most interesting part is this. Some structures called the spindle apparatus, which are these little tubey things here, and these purple dots start to appear. And in essence, we've had to sum it up very briefly. The spindle apparatus is there to move the chromosomes around. Let's jump to this block of text for a second. Earlier in the semester, when I was talking about the organelles that you find inside cells, we spent a lot of time talking about the membranous organelles, but there was also a part about the non-membranous organelles, which are the ones made out of non-membrane material like protein. And at that time, I mentioned that cells had a lot of fibrous proteins inside that form something called the cytoskeleton. These green and red lines are some of these cytoskeletal proteins. And remember, the cytoskeletal proteins are always present inside the cell. They give the cell its shape, for example. And there were 2 main types. The type that I showed you in red is called an actin filament. And the type I showed you in green are called microtubules. And as I said, you find these all the time throughout the cytoplasm of the cell. And in regards to the microtubule cytoskeletal proteins, each microtubule protein is anchored in a structure called a centriole, these little cylinder-shaped objects. And the centrioles are actually made out of microtubules. Each centriole cylinder is made out of a bunch of microtubule proteins, all arranged in a cylindrical fashion. But, as I said, these are the anchors. These centrioles are the anchors for microtubule proteins. Okay, so when the cell is getting ready to divide the nucleus, and that's going to require moving some chromosomes around, some special microtubules are made that are called the spindle fibers. So these are like extra microtubules that are made expressly for moving the chromosomes. And some special centrioles are made for the express purpose of holding those microtubules, so holding the spindle microtubules. And those special centrioles that are made are called centrosomes. Let's click back here. All right. So the purple dotted lines there are the spindle or spindle fibers. Those are the special microtubules for moving the chromosomes. And the little cylinder-shaped things are the centrioles where those spindle fibers are attached to. And those special centrioles that are made for attaching the spindle fibers to are called centrosomes, and each centrosome is one pair of those centrioles. So you find one centrosome that is two centrioles at the north pole of the cell, at this pole of the cell, and you find another centrosome at the opposite pole of the cell, and that centrosome is also made of two of these centrioles. I always kind of wondered why two. What's special about two centrioles? I'm not sure, but that is the number. All right, so the centrosomes, in prophase, the centrosomes appear at the opposite poles of the cell, and the spindle fibers appear and start kind of growing downward to attach